Good morning, good afternoon, and good night to everyone listening to right now. My name is Aryel, and today I will show to you a little about how a leaf cutter ant deals under successive exposures to antagonistic fungi. The leaf cutter ant system is based on a mutualism between the Basidiomycete Leucoagaricus gondylophoros, known as fungus-garden, the ants, of course, and associated microbiota which are beneficial or neutral interactions to the ants and to the fungus garden. Together, they compose an intricate symbiosis that allowed the actors to eat distinct niches and environments throughout millions of years of evolution. However, during their activities to manure the fungal crop, for example, the ants can bring through the leaves or even onto themselves uh, alien microorganisms whose doesn't belong to this symbiosis. Once inside of the colony, these microorganisms can cause antagonistic interactions to the ants, to the fungus garden, or even to the other microorganisms. So, um, how does the system evolve to prevent or diminish the entry and outcoming of unwelcome pathogens? Apparently, the system is well shielded by defensive strategies with chemical, physical, and physiological barriers, which can be applied not only by the ants, but also by the fungus garden and other microorganisms associated. Using these defensive strategies, some studies have shown that leaf-cutting ants can selectively remove these contaminants, depending on the lifestyle or the ecological role of these fungi in the system. After all, it is relevant for the ants to differentiate between potential mutualisms from those with bad history in the colony. However, little is known about it in other leaf-cutting ant species, such as Atta, which is very commonly found here in Brazil and also well studied. So, knowing that multiple and recurrent threats may imbalance in this symbiosis, we inquire for Atta if distinctive fungi trigger distinctive responses in the system. Also, knowing that these ants might be entering in touch through generations to the same pathogens, we inquire do repeated infections to the semifungal strain elicit increased responses in the colony? So for that, we challenge Atta colonies against 5 commonly found alien microorganisms, which is commonly found inside of their colonies and present distinct lifestyles. For example, Trichoderma, Escovopsis, and Syncephalastrum are known to be bad for the fungus garden. Metahesium anisopli is an entomopathogen, therefore it is bad for the ants. And Fusarium oxysporum, which is commonly found in the surrounding soil nearby their colonies. So we don't know for sure if it is a bad fungus for the ants or for the fungus garden. To mimic the successive encounters that the colony might have in nature, we presented each of them in successive encounters in 3 exposures with 14 days apart. We utilize a filtered conidia and with concentration, the same concentration to all of them, and a some solution for the control. To measure how the colonies react under these challenges, we measure their hygienic behaviors commonly applied in these cases. We select the common hygienic behaviors to remove the contaminants from the ants, which is self-grooming and allo-grooming, and to remove those contaminants from the fungus garden, which is fungus grooming and fungus weeding. We recorded the colony 4 times at each interval and later analyzed it and counted the behaviors shown by the ants. Later, we introduced our data in GLMM analysis in our studio with random factor as the colony and the quadrants where we inoculated our treatment, and as fixed factor, the interaction between treatment and exposure. So during the exposures, we could notice that the colonies tended to follow a pattern depending on the inoculated fungus. For example, you can see for Syncephalastrum that even we introducing this pathogen with 14 days apart, the colony not only increased their responses, but also tended to follow a similar pattern throughout these exposures. And you can tell that as well for Metahizium and Trichoderma. Also, it is it's quite different how the ants regulated their responses depending on the fungus. And you can tell that by looking how they react throughout the hours. So it corroborates that leaf-cutting ants might be able to detect and regulate their responses depending on the fungus encounter. In addition to that, the average of strategy applied also varied depending on the fungus. For example, you can see that to remove the contaminants from the ants, they applied more for Metahizium and also for Trichoderma. Even fungus grooming, which is very commonly applied to all of them, you can see that the average also has different strength, with more application regarding Syncephalastrum and Fusarium. It shows to us that it's not only depending on the lifestyle of the fungi inside of the colony, which might be influencing which strategy the ants will apply, but also the context where the spore or the conidia were found. For example, even being an entomopathogen, ants also apply a huge amount of behaviors to remove this fungi from the surface of the fungus garden. Regarding our question about responding to distinctive fungi, what our result shows to us is that the ants tended to apply distinct amount of response. For example, we can see higher and distinct response applied to Fusarium, Metahizium, and Syncephalastrum, with lower and similar response between Escovopsis, Trichoderma, and even the controls. So do you remember as well about Fusarium that I said before? Apparently, this fungi elicit higher responses from the ants to remove it from its colony. So apparently, this fungi it might not be good for the colony as well. However, we cannot exclude other factors which might be influencing which strategy or how much behaviors these ants will apply to remove the contaminants, such as the odor, the germination rate of the spores, how long these spores took to germinate. So we need more studies to unravel that. And at last, sequential exposures apparently incited higher responses, a rapid regulation of them throughout exposures. And you can see that by looking at the first and the third exposures to each fungi, which is not only higher, but also statistically distinct, with differences to Escovopsis, which is only showed a higher increase in the second exposure. So apparently the colony tend to recruit more ants to contamination zones with a previous seen pathogens, with uncertainties to Escovopsis. So with that, it suggests that these leaf-cutting ants might have plasticity under continuous contact to the same and common pathogen that might be brought to the colony commonly several times. Perhaps it can help the colony to remove faster or even in more efficiency these spores before the fungus germinating and causing problems inside of the colony. However, we need more studies to clarify that because we don't know if more ants means more efficiency. So I want you to take home these following messages: that leaf-cutting ants, exclusively Atta sexdens, can detect, recognize, and treat unwelcome fungi species specifically, and that increases their response throughout exposures to the same fungus, suggesting that this colony might be retaining information about previous infections. And what remains to be addressed? What regulates these species-specific actions? It is the odor, the lifestyle of the fungi, or even the germination rate? We don't know. Does this increase contribute to faster and more efficient removal of the contaminants? And do leaf-cutting ants retain an information memory? So who's retaining this information about the past exposures? Is it the ants, the fungus-garden, or the other microbes associated in the system? We don't know yet. So I want to thank you for your attention. Also, the other collaborators, which helped me a lot during this study, in special my laboratory group and my advisor, and the agencies of financial support, which helped me a lot to do this research. If you have any doubts, you can write for me or question me right now. Thank you.